Our next presenter is Ethan Smith. He's a graduate student at Michigan State University. Go Spartans. His talk is on the utility of microbiological profiling for identification of trace soil samples.
All right. Good morning, and thank you, Chris, for that introduction. Before I get started, I'd just like to say it's an honor to be able to present in this session as a graduate student and in the company of people that have been doing trace evidence for longer than I've been alive, and that's a compliment, trust me. And to give you some fair warning, this is a DNA talk. I went through my speech last night, and I think I counted the D word about 13 or 14 times, so bear with me. With no further ado, this presentation is an overview and summary of the soil research conducted at the Michigan State University Forensic Biology Laboratory, which examined microbial profiling for the characterization of trace soil samples. Here's a quick overview of my talk. First, it's important to mention the background and significance of forensic soil analysis, something I'm sure you're all aware of, and the reasoning behind using microbial analysis of soil as an additional tool in the forensic soil analyst's tool bag. The rest of the presentation will be dedicated to discussing the research that was conducted at Michigan State University. The results and conclusions of the different studies will be covered and how each project transitioned to the next. Forensic soil analysis has been employed for well over a century, with one of the earliest documented criminal cases involving the German chemist Georg Popp in 1904. His analysis of the physical characteristics of soil recovered in a murder scene in Frankfurt, Germany, led to the eventual confession of the main murder suspect, Karl Laubach. Soil can be of tremendous value as trace evidence in forensic investigations, wherein questions and known samples are either differentiated from each other or are shown to likely have come from the same location. In fact, soil evidence is most often submitted as an evidentiary versus a known sample. On occasion, although much less often, soil is submitted where there is no known location, and the job for the examiner is to determine where it may have come from, which could help determine locations to further investigate. Traditionally, forensic soil analysis examines physical or chemical characteristics that you see listed here. I'm sure many of the trace analysts here today have examined many of these. Although these can be very discriminating and are still the standard in forensic soil analysis, many require years of training and experience, and some can be very time-consuming. With the advent of DNA technologies, microbial analysis has emerged as a possible way to characterize soils with some of the techniques that you see listed here. Microbial analysis can also be very discriminating, and the techniques do not require as much training to perform, leading to less analyst interpretation. However, some are still time-consuming and variable in their results, and thus are not ideal for forensic purposes. Extensive research into bacterial profiling of soil samples for forensic application has been conducted at the Forensic Biology Laboratory at MSU. The goal of this research was to examine several important factors that need to be determined before microbial profiling can be widely accepted as a means of characterizing soil samples. These include interhabitat variability, which helps determine if habitats can be differentiated from each other, and intrahabitat variability, which can help determine if soil bacteria vary temporally and spatially. All are crucial in a forensic context. If all habitats are similar, there would be no way to determine if an unknown sample came from a particular location. Likewise, if habitats varied spatially and across time, it could not be concluded that a particular soil sample came from a certain area unless that exact location was known. Like all scientific techniques, microbial profiling of soil must withstand Daubert or Frye challenges. Unfortunately, trace evidence is recognized as being highly variable, meaning measurable error rates and reproducibility can be an issue. In this regard, and considering the abundance of trace evidence at crime scenes, techniques that produce reputable results and are subject to statistical analysis are warranted. The methods that will be detailed here sought to meet these challenges, which could prove vital in translating the assays into useful forensic tools. In the initial studies, the effectiveness of terminal restriction fragment length polymorphism, or TRIFLP, was investigated. Myers and Ferran analyzed soils from 5 different habitats around mid-Michigan. The agricultural field was planted in soybean during the summer of 2004, and then in the spring it was tilled, fertilized, and planted in corn at the end of May of 2005. The yard was mowed on a weekly basis during the summer months, while the marsh and the woodlot were undisturbed by human activity during the collection period. The sandy woodlot was also undisturbed by human activity during the collection period and was approximately 100 miles away from the other locations. Soil samples were collected at a main or central site approximately 0 to 5 centimeters in depth at the beginning of each month from September of 2004 to August of 2005. This tested interhabitat variability as well as intrahabitat temporal variability. In addition, soils were collected 10 feet from the main site in the 4 cardinal directions, north, south, east, and west, every 3 months to test for intrahabitat spatial variability. Soil samples were stored in a -20 freezer shortly after collection. Although DNA was initially extracted using the ultra-clean soil DNA isolation kit, successful amplification of all samples was only accomplished using a power soil DNA isolation kit, and this most likely stemmed from the ability of the power soil kit to more successfully remove inhibitors of the PCR and restriction digestion process, such as humic acid. T-RIFLIP has advantages over other techniques in that it provides high resolution of bacteria present in a sample through generation of DNA fragments of variable sizes that can then be separated via capillary electrophoresis and visualized as different peaks on a corresponding electrophoregram, seen here. The x-axis represents the size of the fragment, while the y-axis is just signal intensity. T-RIFLIP is also locus-specific, meaning that each peak could represent a DNA fragment from the restriction digestion of different bacterial species. The universal 16S rRNA gene was targeted, generating profiles that encompassed all bacteria present in the soil. The 16S gene is analyzed extensively in microbiology as it is highly conserved in the microbial genome, yet still has variable regions that can distinguish different bacterial groups. This variability in the genetic makeup is what helps to generate unique profiles. Similarity indices were calculated and examined with analysis of variance and multivariate analysis of variance. A similarity index is simply calculated by determining the number of peaks that 2 samples share or have in common, with 0 indicating that they share no peaks and 1 indicating that they would share all peaks. When determining interhabitat variability, similarity indices were calculated to determine how similar or unique the 5 habitats were to each other during any given month. Habitats were most different from each other in March and most similar in October, and the month in which the habitats were compared to each other made a statistically significant difference in the similarity index. Intrahabitat temporal variability was more pronounced in the spring, likely due to changes in bacterial abundance at different times of the year. The agricultural field, which was manipulated several times throughout the year, was the only habitat to show significant individual changes over time. No significant differences were present when comparing similarity indices of samples collected different distances away from the main collection site. This indicated that time resulted in more heterogeneity than position. Linz and Ferran modified the previous research by narrowing the number of species analyzed to those of the genus Rhizobium, bacteria that are widespread in soil and that require legumes such as peas, beans, clovers to propagate. The goal was to decrease the complexity of the T-RIFLIP profile, and in doing so, increase reproducibility and the ability to differentiate habitats. DNA extracts from the previous study were used to target the REC-A gene. Similar to the 16S gene, the REC-A gene is also highly conserved, but has variable regions that can allow specific bacteria, such as Rhizobium, to be targeted. Non-metric multidimensional scaling was used this time to analyze the similarity indices. NMDS, as I'll refer to it from now on, is suitable for a wide variety of experiments because it does not seek to find any particular relationship between variables, which can otherwise negatively impact the robustness of other multivariate statistical techniques. Data are first ordered into distance matrix, and these distances are then plotted in multidimensional space, in our case 2. Samples that are far apart on the NMDS plot are more dissimilar and vice versa. This strategy showed that several of the habitats could be differentiated. This plot demonstrates the ability of the NMDS to separate the habitats while also incorporating the temporal data. Here, the sandy woodlot and the woodlot show distinct clusters. The plot on the left shows intra-habitat spatial variability for the month of December. With the exception of the yard, habitats formed distinct clusters. Separation of the sandy woodlot, green, and the woodlot, blue, was also observed in the month of June. The fall and spring months did not show as much separation, possibly indicating increased spatial variability. This could be due to anything from differing amounts of human manipulation for the different habitats to geographic distance or seasonal changes affecting bacterial abundance. In pairwise comparisons, almost all habitats showed complete separation. In fact, when an unknown, which was simply just a sample from a different collection period, was plotted with knowns, they grouped with the correct habitat 70% of the time. Here is a pie chart that depicts this. Pairwise comparisons are important to consider because they more closely reflect a forensic situation in which the prosecution argues that the evidentiary sample originated from one location, while the defense argues that it originated from another. Overall, results for this research showed improvement versus targeting all bacteria. NMDS allowed for a visual representation of how samples from different habitats grouped together and demonstrated the usefulness and applicability of pairwise comparisons. Although very informative, T-RIFLIP is not quantitative, and the peaks cannot be attributed to a particular bacterial species, potentially missing important abundance values. Most recently, trace soil research at Michigan State University has focused on the relative abundance of different bacterial groups using a common forensic technique, real-time PCR. Real-time PCR can determine the initial amount of bacteria in the soil and can therefore use abundance of different bacteria to help differentiate the samples. This research also attributed a statistical significance to the patterns seen in NMDS, which aid in the confidence of the results obtained, a feature that is important for Daubert considerations. And finally, most crime laboratories already have real-time PCR capabilities, aiding in the ease of validation and implementation of the assay. New soil samples were collected from 4 different habitats around mid-Michigan. Again, the agricultural field was planted in the summer of 2009 in soybean and was harvested in October. It was tilled, fertilized, and planted in corn at the end of May 2010. Samples were also collected next to a marsh, a yard that was frequented by campers and hikers, and a woodlot that was located next to a hiking trail. Samples were first collected from a main site every few days, then every few weeks, and finally once a month. This again tested interhabitat variability as well as temporal variability. During one collection period in the fall and spring, samples were also collected from different depths in the soil using a soil probe drilled 10 inches into the ground. The core was removed and samples were cut into 2-inch increments. Again, samples were stored in a -20 freezer shortly after collection, and the DNA was extracted using a power soil DNA isolation kit. The DNA was then screened for the presence of different bacterial groups using, again, 16S rRNA-specific primers, except for this time primers were designed with a software program called ARB, which uses a well-established 16S database that contains quality-controlled, near full-length aligned rRNA sequences. These primers were tested with control DNA from pure bacterial cultures provided by Dr. Peter Van Berkom and Dr. Patrick Elia of the U.S. Department of Agriculture's Agricultural Research Service in Beltsville, Maryland. Any bacterial group that amplified in all soil types was then run through real-time PCR. Because real-time PCR was used to assess the relative abundance of the different bacterial groups, and these abundance values were then used for statistical analysis, it was important to find bacteria that amplified in nearly all habitats and whose abundance varied among different habitats. This would allow unique profiles to be generated. With the help of Dr. Tom Schmidt in Michigan State University's microbiology department, Brady Rhizobium japonicum, group 1 acidobacteria, the genus Burkholderia, and the genus Agrobacteria were screened and found to be successful candidates for the assay. I was going to go through each of these one by one, but in the interest of time, I'll go ahead and skip to the next slide. Here is a table showing the different habitats and the bacterial groups found within them. With the exception of the genus Agrobacterium, which did not amplify in the woodlot, all bacteria amplified in all habitats. Seen here are the real-time PCR profiles for Brady Rhizobium japonicum versus Acidobacteria group 1, present in a single habitat, in this case the marsh. Cycle number is along the x-axis, and just relative fluorescence units are along the y-axis. This shows only 2 of the bacteria in the assay, but it can be seen that Bradyrhizobium japonicum amplifies much later than the replicates for group 1 acidobacteria, meaning that it's in much lower abundance. This, in combination with a single bacterium having varying abundances in different habitats, helps distinguish the different soil samples. Many multivariate statistical techniques, including the ones that were used in the previous research, although extremely useful for their purpose, do not rigorously express the nature and degree of uncertainty concerning the data set. To accomplish this, data were first analyzed using multivariate ANOVA based on dissimilarity, or ADONIS. Similar to ANOVA, ADONIS produces a p-value indicating if there is a significant difference between parameters in your data set. Again, non-metric multidimensional scaling was performed. However, in addition, 95% confidence ellipses were also constructed around the data. In regards to this assay, these contribute or attribute a certain level of confidence to the data that fall within them. Interhabitat variability was analyzed using replicates from each habitat from the first collection period. ADONIS was performed, differentiating the data set based on habitat. This gave a p-value of less than 0.05, indicating that at least one sample was significantly different from the rest. NMDS confirmed the results obtained from ADONIS, as the plot on the left shows that the samples from the marsh, samples from the agricultural field were isolated from all the others. This plot is the graphical representation of the data set and shows the confidence ellipses around the samples for each habitat. At first glance, these confidence ellipses do not appear to include many data points, but one must consider that this data set is rather small and that multivariate analysis was only being performed on 4 variables, in this case the 4 species of bacteria. Separation of all habitats should improve with additional samples and more bacterial groups added to the assay. Intrahabitat spatial variability, well, let me go back one, sorry. As seen with Linz and Ferran, separation of habitats was attained more regularly when done in a pairwise manner. The plot on the right shows the woodlot versus the marsh. Although a simplification of the data set, these comparisons more closely, again, represent a true forensic situation. Okay. Intrahabitat spatial variability was analyzed using replicates taken from different depths. ADONIS was performed differentiating the data set based on depth. This was done for the agricultural field and the woodlot in both the fall and the spring. Neither habitat showed significant differences, meaning that the bacterial abundance was relatively homogeneous at different depths, which is good for our purposes. NMDS plots incorporating the depth data for both the agricultural field and the woodlot show that the habitats can still be partially separated and support what was seen with ADONIS. If the habitats were highly variable at different depths, separation may not have been as apparent. Again, this shows the strength of pairwise comparisons. Intrahabitat temporal variability was analyzed using replicates taken from different times of the year. ADONIS was performed, differentiating the data set based on time. Results indicated that bacterial abundance in the woodlot and the yard did not significantly change over time. However, the agricultural field and the marsh gave p-values of less than 0.05, indicating that these habitats did significantly change over time. The agricultural field was a rotation crop, which could certainly change the bacterial abundance at different times of the year. And the marsh consisted of very wet soil that could cause bacterial populations to be more sensitive to seasonal changes, as well as the possibility of the top layers of those soils being washed into the marsh during the spring runoff. These samples, again, were collected from the surface, and that could certainly change the bacterial abundance as well. NMDS plots somewhat support this conclusion, as separation was not as clear when incorporating the temporal data. As you can see on the plot on the left, NMDS was able to separate out the marsh from the other habitats, but separation was not as noticeable when compared to the habitat replicates, which were used to determine the interhabitat variability, which were collected all on the same day. This could be attributed, or the lack of separation could be attributed to the temporal variability of the agricultural field and the marsh, as denoted in ADONIS. Once again, NMDS performed on pairwise comparisons of the different habitats across time was able to separate out most samples. The plot on the right shows the temporal data for the yard versus the temporal data for the marsh. The two habitats, as you can see, the samples generally group with their respective habitat. Here is a table that shows the percentage of pairwise comparisons where complete separation of the two habitats was achieved. This is based on the total number of pairwise comparisons possible for each type of variability tested. The decrease in the ability to separate habitats when incorporating temporal data and spatial data may be due to this assay's increased sensitivity to bacterial changes. In the previous studies, bacteria were also shown to vary temporally and within a single habitat. However, by monitoring relative abundance of these bacterial groups, the assay was likely more sensitive to this variation. Because of this, a presence/absence study may have been more useful. In summary, great progress has been made in determining the utility of microbial profiling for characterizing trace soil samples. The work done by Myers and Ferran demonstrated that targeting all bacteria produced complicated T. riflet profiles, but still showed that samples from the same habitat on average were more similar to each other than samples from different habitats. Linz and Ferran improved the resolution of habitats by narrowing the bacterial target to a single genus, using non-metric multidimensional scaling and making pairwise comparisons. Smith and Ferran saw many of the same trends from the previous studies, but focused on the relative abundance of specific bacterial groups. The addition of Adonis and confidence ellipses helped attribute statistical significance to the trends seen with the NMDS plots. I would like to thank the NIJ and the following people for making this presentation, including the numerous contributors to each project. There would be several slides if I included everyone that helped out with this. I'd also like to thank the Forensic Science Foundation and again the National Institute of Justice for funding a portion of this research. And thanks to the audience for listening to a DNA guy give his side of the story. I welcome any questions after we're done. Thank you.